Peroxisomes are organelles found in almost all eukaryotic cells. Its structure is that they have a single membrane that contains digestive enzymes for breaking down toxic materials in the cell. Peroxisomes are derived from endoplasmic reticulum and replicated by fission. This organelle is surrounded by a lipid bilayer membrane, which encloses the crystalloid core. The bilayer is enclosed with a plasma membrane, which regulates what enters and exits the peroxisome. There are at least 32 known peroxisomal proteins called peroxins, which carry out peroxisomal functions inside the organelle. The organelle's function is that the peroxisomes hold onto enzymes that require oxygen, oxidative enzymes. They absorb nutrients that the cell has acquired, digest fatty acids, and play part in the way alcohol is digested, as well as the digestion of amino acids. Peroxisomes work in a very specific way. Their enzymes attack complex molecules and break them down into smaller molecules. One of the byproducts of digestion is hydrogen peroxide. Peroxisomes have developed into a point where they are able to contain that hydrogen peroxide and break it down into water and oxygen. The water is harmless to the cell, and the oxygen can be used in the next digestive reaction. Some diseases linked with them are peroxisome biogenesis disorders, PBDs, which include the Zellweger syndrome, chondroplasma, and rhizozelmic puntia type 1. They represent a continuum of disorders including neonatal adenocarcinoma, infantile rufum disease, and Zellweger syndrome. Collectively, BPDs are autosomal recessive developmental brain disorders that also result in skeletal and craniofunctional dysmorphism, liver dysfunction, progressive sensorial hearing loss, peroxisome biogenesis disorder. This results in the overaccumulation of very long-chain fatty acids and branched-chain fatty acids, such as phenolic acid. In addition, PBD patients show deficient levels in plasma genes, either phospholipids necessary for their normal brain and lung function. These diseases are caused by the mutations of the PX7 gene, which encodes that the PTS2 receptor. Patients can develop large tissue stores of branched fatty chain acids such as phenolic acid and shows reduced levels of plasminogens. There are no known cures for any of these diseases.